Good morning, everyone. Myself, sneha Purbia from Group 316. Here is my topic electronic conformation interaction in proteins and cooperative properties of enzymes. Let's get started with the topic electronic conformation interactions and proteins. First, what is electronic conformation interaction in proteins? It refers to the influence of the electronic structures on the spatial arrangement of atom within the proteins. These interactions are very crucial for understanding the protein stability, protein folding, and protein functions. Let's start with here. The functionality of a biopolymers and the supermolecular structures in the biology strongly depends on the interaction between the electronic excitations and the shift of the electronic density with the conformational changes in the system. The study of the electronic conformational interactions can help us to form the physical theory of the enzymatic activity and other biomolecular phenomena. The concept of the conformation can be introduced for the theoretical description of electronic conformational interaction, describing a kind of a quasi particle containing the shift of the electronic density and the conformational changes. The direct experimental investigation of the electronic conformational interaction were performed with the system known as apo-aspartate aminotransferase system with a series of different coenzymes. The denaturation of such systems with that is the conformational properties changes. This is maybe due to different reasons like temperature changes, pH changes, or any another factor will affect strongly on the kind of their electronic interactions at their active sites. So let's start with their introduction. The living organism and its functional system, including the molecules of proteins and the nucleic acids, are complex chemical machines characterized by the self-consistent, regular, and regulated behavior of their elements. The main function of a biological system are chemical, that is participation, particularly primary the catalytic one in electronic transformation and the realization of the electronic and ionic motions. The motion of the electron in the molecules is coupled with the nuclear motions. The nuclear movement in the biopolymers are specific. The particularities of the structure and properties of the protein molecules are determined mainly by their conformational mobility. The analysis of the electronic conformational interaction must lead an explanation of the main features of enzyme catalysis. On the other hand, the behavior of contractile properties responsible for biological mechanochemical process such as muscular contraction follow also the transformation produced by some electrochemical phenomena. So let's start with the enzyme catalysis. So to explain these enzyme catalysis, we have a theory known as a Koshland theory, which played a great role in development of a modern theoretical treatment of enzymes, support the great importance of the transconformation for electronic process catalysis by the enzymes. However, this theory consider only static pattern of induced structure fate of enzymes and substrate, but not the dynamical phenomena which determine the formation and further fate of the enzyme substrate complex, also known as ES complex. Uh, let's understand the experimental study of the electronic conformational interactions in the protein. By the rise of the spectacular model of interaction between electrons and the atomic nuclei, we can understand how this electron help us to understand this concept. Um let's start. Electron in a partial box with infinitely high mobile walls. This is a diagram to explain this theory. This is a potential box. Here we have a different levels. N is equal to 1, n is equal to 2, n is equal to 3. In this level, the electrons are present. If any changes occur, it will reflect in this graph. So let's start. Uh, First, the electrons are placed in a blocks where 2n electrons occupy in a n levels. Energy of the electron inside the box is calculated by a de Broglie standing wave formula, which is lambda is equal to 2L by n, where L is the width of the box and n is equal to 1, 2, 3, 4, etc. Velocity, by this formula we can also uh, find out the velocity of electron in the box by the formula v is equal to h by me lambda, where h is refers to the Planck constant, me is the mass of electron, and lambda is a de Broglie constant or de Broglie wavelength. Let's put this value of the lambda and we got this formula, nh upon 2 mel. From this formula we can also find the kinetic energy, that is mev square by 2, that's equal to n square h square upon 8 mel square. By this formula is come out by putting this v value. We can also find the electron exert a pressure on the wall by this formula. F is equal to differentiation of E upon differentiation of L, and we got this formula n square h square upon 4 mel cube. Let's see the changes in this graph. First, the n is equal to 1, the graph is come like this, but if we change the value of the n, the graph will change like this, the dotted line. Learn by the theory. At equilibrium, the force are compensated for by the interaction with the surrounding of the box. The change in the equilibrium results either from the excitation of the electrons uh, um, in the system or from a addition of a new electrons in the system. Uh, uh, Either of these leads to the increase in the pressure in the partial box. It can also explain the distance increase in this partial box. At the equilibrium, we know that the wall of the box are moves, so it will induce in a change in the length, a change in the distance. So we can say that the L plus change in length is our actual length in the of the box. This also explain the electronic energy decrease with the increasing in length. We know that the energy is equal to n square h square upon 8 mel square. As the length increases, the energy is decreases. Important objects for the study of the electronic conformational interactions are metallocoenzymes, in particular enzymes whose cofactors are ion of transitional metals. Here we have a graph which are showing the conformational electronic conformational transition. Here the on the x-axis we have Q, on the y-axis we have U. and this graph showing the electronic conformational transition, and these also explain at the equilibrium state. So let's get started with another topic, cooperative properties of enzyme. So what is the cooperative properties of enzyme? It is a enzyme kinetics that refers to the phenomena when the binding of a substrate molecule to the one subunit of a multi subunit enzyme influences the enzyme affinity for additional substrate binding. The cooperative properties of the enzymes, particularly focusing on the differences between the Michaelis Menten kinetics and the kinetics of the enzymes that exhibit the cooperative behavior. Introduction to the cooperative kinetics. The kinetics of enzymatic reaction can shows the complex behavior deviation from a simple hyperbolic shape predicting by the Michaelis Menten kinetics. Instead, the reaction velocity V may display the inflection, maxima, and the plateau depending on the substrate concentration and the modifier concentration. Let's take an example of a aspartate transcarbamylase. Um it represent the presence of the CTP, cytidine triphosphate, modifies the reaction curve, shifting it from hyperbolic to the sigmoid when the CTP is absent. Let's see in the graph. We have a velocity on the y-axis and the substrate on the x-axis. We know that if the CTP is present, then the graph is come the sigmoid. But if we remove the CTP or the CTP is absent, the graph will come hyperbolic. It explains the how these enzymes affect uh, the presence of the CTP and the not CTP present. So let's start with the enzyme structure and the cooperativity. The cooperative behavior of enzyme is typically attributed to their quaternary structure, and the interaction between the subunit, that's the promoter. Each subunit can have its own active sites, and the binding of substrate to one active site can influence the activity of the anothers. Uh, we have models to explain these subunits. Uh, Enzymes with the two identical subunit existing in a three sites. First, the both sites are free. The second is the one site is occupied. Third is the both site is occupied. This. Um, we can say that the possible structure of enzyme for the isologous means the same enzyme is bind to each other, or heterologous different enzyme bind to each other, or dimers of the heterologous polymers with many heterologous enzymes bind each other. uh, then then study the models of the cooperativity. We have two models for the cooperativity. First is the direct cooperativity model, and second is indirect cooperativity model. Direct cooperativity, um, it involves the different rate constant for the enzyme state when the substrate occupies a one or two active sites. Second is the indirect cooperative model. It is proposed by the Monod, Wegman, and Changyang's. Uh, Changyang's. It also known as the MWC model. It suggests that the enzymes can exist in a multiple conformational state with the substrate. Binding shifting the equilibrium between these states. The uh, this para explains the possible states of the dimeric enzymes and how these can relate to the ligand binding. uh, Allosteric enzymes regulations. So first we know that the allosteric enzyme, and then we study the how this allosteric enzyme affect the cooperative behavior. So what is allosteric enzyme? First, allosteric enzymes are the regulatory enzymes It's a definition, a type of enzyme regulation by the reversible non-covalent binding of regulatory molecule to the enzymes. It is a regulatory molecule that are also known as allosteric modulators and allosteric effectors. Allosteric enzymes has additional conformation induced by the binding of the modulators. Conformation change induced by the allosteric modulators can produce more or less active form of the enzymes. We have the structure of enzymes. Here we know we have an active site where the substrate can bind, and we have a allosteric site another than an active site where the inhibitors can bind, or we can say that the non-competitive allosteric inhibitors can bind. So on the basis of the nature of these modulators, we classify the allosteric enzymes into two different parts. First is the homotrophic allosteric enzyme, and second is the heterotrophic allosteric enzyme. Homotrophic allosteric enzyme. If the substrate act as a modulator, that is the allosteric enzyme having the substrate and modulator are same, are called as homotrophic allosteric enzyme. Example is a hexokinase. Second, the heterotrophic allosteric enzyme. If the substrate does not act as a modulator, that is, if the modulator in any molecule other than substrate, the enzyme is called as heterotrophic allosteric enzyme. Its example is the threonine dehydratase. So, see how this allosteric enzyme affect the cooperativity. These allosteric enzymes exhibit cooperative behavior and are regulated by feedback mechanism. For instance, aspartate transcarbamylase is inhibited by the CTP. The end product of the metabolic pathway is catalysis. This feedback mechanism ensures that when the end product accumulates, the enzyme activity is reduced. It prevents the overproduction of these enzymes. Then let's study the kinetics of the allosteric enzymes. Uh, it explains the allosteric enzymes displays a quaternary structure which allows them to associate it with the subunits under the influence of the substrate and the allosteric effector. The kinetics of these enzymes can be modeled mathematically to illustrate how they can respond to the substrate and inhibitors, showing different behavior compared to the typical Michaelis-Menten kinetics. Example, let's take example of threonine deaminase. The kinetics of the conversion of the threonine by these enzymes may be described by the formula, that is the log V upon V max minus V is equal to n log S minus log K, where this V is the velocity, V max is the velocity, maximum velocity, and uh, n is equal to n 1 2 3 4 etc, and S is the substrate concentration and K is a constant. And the kinetics of the allosteric inhibitors dissipated as follows: log V is upon V naught minus V is equal to log K dash minus N dash log I. Here sorry, V naught is the velocity at I is equal to 0. Uh, for the Michaelis-Menten kinetics, n is equal to n dash is equal to 1. But Changiax found that the n is not always 1. N is equal to 1.37 and n dash is equal to 1.86. This result easily explained by cooperativity, the interaction of the subunits. Now let's explain the relaxation kinetics. It is the final section that delves into the relaxation kinetics, where enzymes are studied as they return to the equilibrium after perturbations. This approach can reveal insight into enzyme dynamics and cooperative behavior, even in the system where the cooperative interaction may not be overly evident. So, this is our topic, cooperativity in the enzymes. Thank you.